a lot of questions for Dominique. I would start with a question from Clay Wright, and they ask, how can we separate biological noise from true differences that are meaningful to development between precursor cells?
That is an excellent question that I will not say we've solved. I think we've revealed it's interesting in development. The closer you get to the endpoint, to the differentiated cell, the cleaner the signal is. So guard cells, we absolutely know what those are. Those precursor cells, there's variation in there, and determining what is noise and variation is definitely a challenge. I think what's very nice is that this is actually a general problem, not only in our data, but everyone's data. And so if you have 500 human cell biologists with the same problem and their resources, we can actually tag onto that. And so there are people trying to come up with algorithms for those. We're trying them, but it is definitely an ongoing challenge.
Okay, thank you. The next is from Heather Meyer. How conserved are the stomata lineage proteins across plant species, and how well can the single cell data in Arabidopsis will help us to determine what is occurring in other species?
Yeah, so that's the key question. How much can we port what we know in a model species out to other ones as a general question? And again, the closer you get to a terminal cell type like a guard cell, I think it's pretty straightforward. And it's not just one gene. The thing is that you're relying on groups of genes that are behaving in similar ways. And so this is something that you see in other cell types as well. So I have a great deal of confidence we'll be able to figure out what guard cells are in many species. I have much less confidence that we'll be able to figure out what precursor cells are because the noise and possibility, and we don't know, we still have to functionally validate a number of those in model species. So I think that's a really important thing in thinking about when do we go shallow, when do we go broad, and what questions can we ask across species.
Thank you. The next one is from Rajveer Singh. How do we arrest a cell trajectory to study the transcriptome of each stage and other products?
So I think there's two strategies. I mean, one thing is to say, and this is a strategy that Keiko Tori took in the past that was quite successful, saying let's use mutants so that things don't go all the way through a pathway and they accumulate. And when we didn't have the ability to actually capture single cells but had to do it in bulk, that was a good strategy. Now I actually think that's bypassed by the fact that you can separate cells out, you digest the cell wall, you can FACS sort them so you get individual cells, and the technology to be able to capture the transcriptome of individual cells is good enough, or of individual nuclei, that we don't have to arrest cells anymore. We can keep them a bit more in their natural state.
Okay, and
one more question, please.
One more, okay. So the last one is from Jiang Wang. Can you recognize the transcription factors driving development by their spatiotemporal behavior in the data?
That's another goal and Camilla's trajectory analysis to say, what are the transcription factors that would be important? So if you've got something that turns on and you know its targets, you want to know kind of what was the thing that drives the next transition. And so far we don't have any that we know are doing that, but that's an active area of research because of course we'd like to be able to capture all these different stages and ask what brings one to the next. And that's probably transcription factors.
Thank you so much. So we can move on to questions for Uta. And so moderator for Uta, please.
Oh, cool. Hey, everyone. So the first question is from Lisa David. How can we move from just transcriptional analysis to talk about the products of transcription, like proteins or metabolites? As we know, many transcripts are degraded or do not make a product.
Yeah, so we actually had already a chat exchange with Lisa a little bit further in the meantime. So thank you very much for that question. As I mentioned first, it depends really on the type of question that you're specifically addressing. So if you're interested in the proteins and metabolites, then, as you quite rightly say, just merely trying to link this with transcriptomics might not be enough. So it really depends on, I would say, without burning my fingers here with any particular strong statement, it really depends on the system you're working with and on specifically the type of question you're addressing.
Cool. Thank you. The second question is from Marcia Pierce. So what is the fate of all the membrane remnants after collapse, and how is plasma membrane integrity maintained?
Yeah, so we know from our electron microscopy efforts that the periavascular membrane actually stays integer until the fungus has entirely, the avascul has entirely left the cell. And there is great discussion about what is actually happening to this huge amount of membrane material that is generated in the course of the development of the avascul. There are contradicting opinions. One report which says there's a lipid burst occurring as the avascul starts. Certainly there is beta oxidation at big time going on to degrade the membrane material. But how you can possibly imagine these huge amounts to be channeled back in both organisms into the regular metabolism is not understood.
Cool. So the next question is from Alexander. Do all AMF symbiosis form in inner cortex, or is there variation between plant species? Like, where is it already known to be a variation in number of cortical cell layers?
Yeah, so as you could maybe see on this gas reporter line slide that I showed, so the avasculs eventually start to form in the inner cortex, but can also form in outer cortex. So what we observe is that they first are developing in inner cortex cells, and then with progressing time eventually also go into outer cortex cell layers. Now, if you have a plant species that is mycorrhizal but doesn't actually have that, might not even have a root. So if you look at liverworts, some liverworts are mycorrhizal competent. So there you would have the avasculs forming in the thallus, so a completely different type of tissue.
Thank you very much, Liwen. That's time, and we'll move to questions for Liwen. I think I said Liwen instead of Uta. I'm sorry. So, questions for Liwen. Hi, Heather.
Okay, great. Hi. So, Liwen, your first question is from Lisa David, and it is, do different cell types have different vacuole development? So, for example, do guard cells have specialized vacuoles?
The answer, I think we don't know yet, but my feeling is yes. That's why we're looking at different cell type for the vacuole development. Root, also the stomata cell, yeah.
Okay, and then on a similar vein, I'll ask a question. Does vacuole composition change depending then on cell type or life stage?
I think so, because we started the protein trafficking to vacuoles. I think when you become more mature, more content will be transferred to the vacuole, okay? Also depends on cell type. I mentioned earlier, in the protein storage vacuole, the normally contain the storage protein rather than the hydrolytic enzyme. So, yeah.
Great. So this is a sort of a joint question between Lisa David and Naomi Nakayama of, well, first there's a comment of, your images are so impressive. Can you please share with us how you captured your images and which software you used for the 3D modeling? It's really amazing.
Okay, we learned this technique. Actually, we have a collaborator, Dr. Kong. I think the other expert in the world is Marisa. I think she's in the audience. I think these are the typical technique called tomography. Basically, you need to have a powerful TEM, okay, the 200 kV, so they can then go through the 6 section. Okay, we cut multiple sections, 300 to 5 nanometer thick, and then we collect serial image, and then the software comes with either with the TEM or there's an open source software.
Okay, great. So this is a joint question of David Earhart's and Sury Jha. So cell size, the driver vacuole size, or is it the state of development?
So that's the question we don't really know. That you know, you have the chicken first, the egg first. There's something like this kind of question. This really is something hopefully the community or we could find out in the future. Yeah.
Okay, great. So we have another question from Rajveer Singh of what are there other cellular factors responsible for vacuole size?
Okay, I mentioned one example, the S-chromosome machinery. Actually, there's so many mutants. There's serial lethal, okay, the snare protein, the S-chromosome machinery, some of the lethal, we look at the mutant. Normally, the vacuole, they cannot go to mature. They're very small. So that maybe could be, you know, that if the cell, the plant cannot get the mature, the large vacuole, maybe they cannot survive. That could be
Okay,
one
great.
of them.
So you have another question from Jiang. Do stem cells have smaller vacuoles than their differentiated derivatives in the root?
That's something we try to look at. The preliminary information, I think so. That stem cell normally the vacuole are very smaller. Yeah. That's something I try to ask my students. We should maybe look at that stem cell. Yeah.
Great, and you have.
That's time.
Oh.
Yeah, thanks though. Great. That was great. Questions for last speaker, for Martin, please.
Hi, my name is Jason Thomas. I'm a postdoc in the Relab at Carnegie, and I'll be choosing some questions that were brought up in the chat. So the first one is from Luke Nikolov. How different are algopyranoids from the pyranoids of hornworts, the only land plant group that has these structures?
Thank you for the question. So essentially, morphologically, at a big picture level, they have a number of similarities. So they still have a matrix and they still have tubules that traverse them, membranes that traverse them. But there's increasing evidence now that pyranoids in general are the result of convergent evolution. So at a molecular level, we expect that they might be different in many ways other than sharing rubisco.
Thank you. Then I have a related question from Chan Yul Yu asking why algopyranoids are lost in higher plants. Can we apply algopyranoids or bilinker proteins to C3 higher plants to saturate carbon dioxide and improve the efficiency?
Yeah, so certainly, pyranoids, we think, are a late evolutionary innovation in certain lineages. And so I currently think that many land plant lineages, basically land plants originally did not have pyranoids, and hornworts evolved them over time. And independently in different algal lineages, pyranoids evolved as well. And so it is indeed our dream to understand a pyranoid at a level sufficient for engineering it into C3 crops like rice and wheat to increase their yields.
Thank you. Next question was from Dominique, and she asked, since the pyranoid is a dynamic structure dependent on carbon dioxide levels, do you have an idea of whether the linkers get altered in these situations?
Yeah, I think this is a great question and certainly something that we're very interested in understanding. It looks like some of the structures stay pretty constant no matter what the environment does, but certainly the pyranoid matrix dissolves under certain conditions and then condenses under others. And so we hypothesize right now that the linker protein that links the rubiscos together may be post-translationally modified to promote condensation or dissolution. But at this time, we don't know how that works.
Great. Next question is from Bakash Raul, and he asks, how exactly pyrenoids would help increase yield in land plants, and where will it be positioned inside the cell? And I wanted to add something to that. What are the main bottlenecks and challenges in engineering pyrenoids into crops?
Okay, so the first one is how will it improve yields? Essentially, it will do two things. First of all, it will force feed Rubisco with CO2, so it will make Rubisco run faster. Second of all, it will increase the amount of CO2 relative to oxygen. And so this will decrease this unwanted reaction that Rubisco does with oxygen. It will favor the reaction with CO2. Second one is where will the pyrenoid be within the land plants? It will go inside the chloroplast. So each chloroplast will have one pyrenoid. And your question, Jason?
Yeah, what are the main bottlenecks and challenges in engineering pyrenoids into crops?
I think the biggest bottleneck right now is still that we don't understand enough about the basic molecular biology of how a pyrenoid is assembled and functions. I think there may be a few components that we still have yet to discover. In particular, right now, the tubules, the membrane tubules that traverse the pyrenoid, we have no idea. We don't know any protein that actually is responsible for forming the tubules. So we need to understand how those tubules are made and actually what they do really before we can engineer them.
Okay, great.